Today it's my great pleasure to chat with a friend and scientist who introduced me to the concept of hormesis, although actually I knew a little bit about it, but based on something else that maybe we'll talk about called preconditioning. And my guest is Professor Edward Calabrese. Ed, he's professor of environmental health sciences at the University of Massachusetts Amherst. He trained as a toxicologist, and he's the world's expert on what's called hormesis, and he'll define that and tell you what it is. He's well known by policymakers in the EPA and maybe the Atomic Energy Commission. And he was, in 2009, he was awarded the Marie Curie Prize for his work on this concept of hormesis, why it's so important, not only for environmental science, but biology, evolution, pretty much anything life scientists study. There's implications for their work, I think, and it's good for them to have this concept in the back of their mind. So welcome, Ed. I've known you for a number of years. Actually, I think, so I read, you had an article, short article in Science back in the early 2000s, where it was something like scientists rethink the core concepts of the EPA, of toxicology or something. And then I can't remember, I think you contacted me or I contacted you, but we got together and started, as scientists do, kind of brainstorming.
Well, thanks very much for that intro. Actually, yeah, it started, well, just on the hormesis side, the audience might be interested in knowing sometimes you have an idea and sometimes you're in love with it, but nobody else is really in love with what you're doing, and a lot of times you face rejection with grant proposals, and many times you face rejection with journals that trying to submit papers to them and scratching your head and trying to figure out a way to move forward. And sometimes it's and you think you might be right, but just because you think you're right doesn't mean that you're going to be successful. And I remember it was, I started on this concept of hormesis a long time ago when I was an undergraduate back, I hate to say this, but back in 1966, if anybody can remember that far back, but and got, and did my master's thesis on it, actually. And then that was in the plant world, working with peppermint and a growth inhibitor. And to the shock of the professor, our peppermint plants were stimulated at very low concentrations due to a mistake that I made in making up the stock solution. How potato chips were discovered, they were just by an accident. And so I followed it up, and this guy was very demanding man, but this was very significant for me because we had a stimulation, and the stimulation it was so different than what he was expecting, which was pretty strong in inhibition. But the stimulation was only about 50% more than the control group. It really wasn't much, but it was a stimulation, and it did surprise him. And so he said that he wanted somebody to figure it out, and I volunteered to do it. And I went back and designed a study and conducted it, and I got the about 30 to 50% stimulation again at low doses. But so what happened is that he wanted for me to see the entire dose response continuum or spectrum from low to very high doses, that the whole thing. And so when I did the whole thing, my goodness, I found that at high doses, I saw exactly what he said we would get, and what prior students had was the plant growth was very inhibited in a dose-dependent sort of a way. But as the dose was kind of titrated down, there was a flip to the other side, and the plants grew more. but it was still only modest. It was, 50%, 60%. And so, and so he said, well, you have to put more doses, you have to have more plants, you have to make it stronger, because you have to convince others about this. And so the bottom line was that I went through with this guy about 12 different replications, adding more and more, however this sequence was, thousands of plants ultimately being tested with this. And also we tested it over time. And so, and one of the interesting things that that emerged on that was that in fact, if you followed it over time, most of the doses, even those in the very low dose range, it was they showed a dose dependent response. There was inhibition even at the low doses. But when you followed them beyond the first and second weeks out to the sixth or seventh week, you saw that the low doses began to be stimulatory, and there was like an overcompensation response. And still the high doses, they were just toxic, and they never really fully recovered at all. And so I had multiple things going on here. I had this high dose inhibition. I had a stimulation, but the stimulation wasn't occurring right away. It was taking time, but it did always occur that way. It was happening in soil. I didn't know what was actually in the soil. Maybe there was some bio, interactions with the chemical and the and fungi or bacteria, changing it to something. He suggested that, well, why don't you do this whole thing in hydroponics? See if you can get the same effects, avoid the soil. And I said, well, that's a great idea. Wished I had thought of it. But so I went back and learned how to do hydroponics, and I did the study, and lo and behold, I got this same low dose stimulation, high dose inhibition, the same phenomenon. This time he only had me replicated about 5 or 6 times, but the results were very consistent. We published it in a British plant physiology journal, and for me, I was so I was beaten down actually by the enormity of the work, and I, the need to replicate and to prove. It was very demanding. And I was I was 20 years old, 21, 22, maybe 20 when it all started. And I got a real taste as to the demands on scientists to actually prove something. And to prove it to my advisor, and it was the 30%, 60%. It was taught me a lot about background variability and control group variability, and how you could get snookered into believing something that you wanted to believe, but it really wasn't true. And how could you get past that? Because he said you have to be your own worst critic. Because you're going to be going out in the world and trying to prove something, and you have a reputation, you have a career. You have to be very, very tough. And you are the sentinel for the scientific community, so you owe them rigor in everything that you do. And I'm 20 years old. I'm saying, well, is this how it is for everybody who goes into science? And actually it should be. And I find that one of the issues with hormesis and so forth is that you really have to see how difficult it is to prove, when you have only and this was also interesting too. I didn't really understand that it was very general phenomenon. I knew that it occurred in these peppermint plants and that it occurred with this chemical that treated them, but did it occur way beyond? And this was at a time, 1966 and 7 and 8. And it was at a time, we didn't have computers. I had to go to the literature, and everything was, like biological abstracts or chemical abstracts.
Oh, yeah.
Everything was done by hand. How do you find information? It was very different, back in the day.
And I had kind of a similar experience, except with a role reversal.
Oh, okay.
And where I was the mentor, and I had actually it was a postdoc. So this is back in the early 90s when I was at University of Kentucky. And we'd been studying this phenomenon called excitotoxicity, where neurons can be stimulated too much by glutamate, an excitatory neurotransmitter, and that can kill them. Low concentrations of glutamate is good for neurons. High concentrations is bad. And then I had this postdoc look at a number of different neurotrophic factors to see if they could counteract the excitotoxic effect of glutamate. So we had cultured neurons we take them from embryonic rat brain, put them in culture. They start as spheres, then they grow processes. One becomes the axon, the other's dendrites. They form connections. They can be stimulated by glutamate at physiological levels, or you can kill them. But so this postdoc found that several neurotrophic factors, proteins that are known to promote the growth and survival of neurons, could counteract the adverse effects of glutamate. Okay, so we had those data, published those, that work was kind of straightforward, got accepted. And then I said to the postdoc, his name was Bin Chang, I said, Bin, let's look at, there's a lot of emerging interest in inflammation in the brain, in Alzheimer's disease, Parkinson's disease, and the notion that chronic inflammation is bad or inflammation is bad. So I asked Ben to do the same kind of design experiments he did with the neurotrophic factors, but with pro-inflammatory cytokines, tumor necrosis factor, interleukin-1 beta, and he did different doses. And, the expectation was that these pro-inflammatory cytokines are going to make things worse. They're going to make neurons be more vulnerable to excitotoxic damage. And there's this emerging evidence from my lab and a couple other labs that this excitotoxicity process is playing an important role in stroke and maybe even in Alzheimer's disease. Okay, so he got, he showed me the data from the first experiment with TNF, tumor necrosis factor. And he found at lower doses, it was protecting the neurons from being damaged by glutamate. It was having a good effect in this model system. And so I did to him what your mentor did to you. I said, okay, this doesn't make sense. That we think I thought, inflammation, the hypothesis was inflammation is bad. So I said, repeat it. He repeated it multiple times, and so then we submitted the papers for publication to actually pretty high impact journals, because we thought this was a very important finding, a good effect of this pro-inflammatory cytokine. And we couldn't get into the top journals. We finally got it into a good neuroscience journal, but not top one. And then we started asking what the mechanism is. How is TNF protecting? And it turns out there's this transcription factor called NF-kappa B that had been shown by immunologists and people who study inflammation to be involved in inflammatory processes associated with inflammatory process. So then I have another postdoc look at this NF-kappa B, and lo and behold, if we activate NF-kappa B, TNF will normally activate NF-kappa B. That promoted survival of the neurons. Okay, and so and same thing happened, repeat the experiments, submit it to high impact journals. It was at Science for a long time. It got initial reviews back, and they gave us opportunity to resubmit. Took another 9 months or a year. Then we finally got the paper in PNAS, take home message, activation of NF-kappa B is good for neurons, promotes their survival. Then 1 month before our paper came out in PNAS, 2 papers came out in Science from immunology groups, including one who was a Nobel Prize winner, was the head of the lab. And they're showing in immune cells and some other cells that activation of NF-kappa promotes survival.
Oh, interesting.
So these are the kind of situation we run into, Ed. So then the obvious question in our minds was, we had submitted our paper to Science. It didn't get in there. The take-home message was the same, the kind of the major take-home message. So what would you think? And then, you know, a month before our paper came out in PNAS, 2 back-to-back papers came out in Science. What would you think?
Well, you know, it's such a strikingly important development. And,
Well, you know, you wonder if it's a coincidence, whether, you know, who was reviewing our paper, was it?
yeah,
Yeah.
yeah, it's hard to, when you're not inside the system to understand what's going on. I find that my imagination runs wild, you know, and it's you know, I tend to think that they're communicating with each other, because if something's exciting, people talk, and so they feed into some sort of common pathway up to some editor that has some commonality with that. And so you'd like to have been like a fly on the wall to try to understand the internal politics, and how that goes on, and you know, just having been it's not the same thing, but I've tried to reconstruct the history of let's say the discovery of mutation with Hermann Muller, and followed his work and his discovery, and there was other people that knew that this concept was important, just like you knew this concept was very important, and that you know, as it turns out, you know, in his case, some people, you know, held on to data for like 4 or 5 years. Then they ultimately submitted it to and got into PNAS 6 months before Muller did, and they were very hesitant, but they wanted their concept out, and they wanted to introduce the concept if they had data. And when Muller was aware of their data, when he published his groundbreaking paper in Science, surprisingly or shockingly, he never showed any data. He discussed his data, and this was extremely odd. And then people who were chasing him came in with data 3 months after Muller, but yet Muller got the prize. And Muller, you know, they didn't know whether he had it or not. How did he get his paper in Science without data? And then he goes to Germany maybe 4 months after the Science paper, shows his data at the conference, and the conference, and publishes his data in a non-peer-reviewed conference proceedings. And you know, whatever the science and how it evolves is it can be very complicating, and why certain papers get in and by whom and how quickly, and I think that there are many stories that remain to be told of the actual human dynamic, and at the same time there's competition, there's desire for prizes, there's recognition issues, and Hermann Muller was certainly, you know, aware of all of those in spades, but it's part of human nature, and so it plays into discoveries today as it did 70 years ago.
So you and I both give examples of where, you know, exposure of some, you know, animal, plant, doesn't matter, cells, to some challenge, you know, that I'm often using the term challenge instead of stress now, because stress is like always, the connotation is stress is bad. But with the hormesis principle, low levels of stress can be good. Exercise is like the classic example everybody knows about. When you exercise, your muscles are under a lot of oxidative stress, ionic stress, metabolic stress. And, they respond over time, with intermittent exercise in ways that make them stronger, more resilient, better endurance. And another classic example is the immune system, right? You're exposed to a virus or a bacteria, at relatively low level initially, your immune system responds. And all of these hormesis-based systems are evolutionarily important. There's this, the saying that nothing in biology makes sense except in the light of evolution.
That's correct. Yeah.
That's Theo Dojansky.
Right.
And then I would say nothing in evolution makes sense except in the light of hormesis. So let's talk a little bit about that. These systems evolved, whether it's the plant or us, bacteria. All these organisms evolve in relatively harsh environments in many ways, where they're subjected to intermittent challenges or stressful situations, for example. And you and I mean, you've written, I should say, if viewers or listeners don't know of Ed's work, he has looked at tens of thousands of scientific journal articles and dose responses in those, and over the whole expanse of the fields of biology and even some in medicine. And so he knows this more than anybody, and have pointed this out. These low dose, stimulation or beneficial effects, high dose, this biphasic dose response. And you've written on evolution too. So one thing in evolution, so it's thought that the first organisms that arose on Earth were actually in the oceans, in the deep ocean, and these like volcanic vents. They're generating a lot of hydrogen sulfide gas, and there's actually very high concentrations of iron, ionic iron. So hydrogen sulfide gas and iron are toxic in high doses, even to humans now. And this is one example of a remarkable evolution, with the hydrogen sulfide gas, a toxic gas and a toxic metal. The bacteria actually, not only did they develop molecular mechanisms to resist the toxicity of the iron binding proteins are ways to move the iron around, and same with hydrogen sulfide gas, but they actually evolve ways to use these toxic substances to their advantage, right? So iron in humans, right? In hemoglobin, very important in getting oxygen moving around to all your cells. But we and other animals evolved iron binding proteins that protect us from the toxic effects of the ionic iron. So anyway, I'd like you to talk a little bit about your thoughts on, kind of from an evolutionary perspective. I think there's a very important concept that people need to get in mind, and including in neuroscience field, which we'll get to.
Well, the most astounding thing for me in terms of discovery, put it that way, in terms of hormesis, and it addresses the evolution question. And that is that, when I started this, I kind of gave the audience a feel for a young kid taking a look at a peppermint plant and watching it being stimulated, and the professor not being terribly impressed with a 50% increase over the control group. But nonetheless, that's what I kept finding over and over and over again in these little studies. Well, 20 years later, I after my PhD, and I wasn't interested in hormesis for 20 years. I was developing my career as a as a biochemical toxicologist, actually.
Yeah.
And then I got reintroduced to this concept of hormesis within an environmental framework, and but I knew that it wasn't really well understood or accepted. It had a couple of proponents here and there, but it had nothing that had made it into really central core science, put it that way. And so I as you mentioned, I began a quest to take a look at the how widespread this was, and the interesting thing is that I found that this hormetic response, this low dose stimulation, high dose inhibition, actually occurred from bacteria to humans, and was in plants, it was in algae, it was at all different levels of biological organization. It was at the cell level, it was at the organ level, the organismal level, and one of the things that was very weird to me, because even though when I had a problem with the professor that it was only like 30% or 40-50% stimulation, very low. When I 20 years later, when I began to think, I said, well, gee, maybe this hormetic response, when I started developing my database, I assumed that it could be as large as 400%. I thought that the stimulatory response could be not unlimited, but considerable. 400%, 500%. When we made our database, we had entry criteria that would allow for that. And so when I began to look at these biphasic dose responses in all systems from all different chemicals and physical factors, radiation, whatever, the very interesting thing that emerged was that we found lots of examples, but the maximum stimulation was almost always very limited to less than twice what the control group was, and the maximum being about 30 to 60%, 70% in that range, greater than the control group. And this was for all life, for all chemicals, and that really meant for probably all diverse mechanisms. And then what got me into further thinking was that I knew that sometimes you found stimulation when you gave a chemical or stress, or however you wanted to look at it, to a system. There could be a direct stimulation. It would directly stimulate almost immediately after it was exposed. But then I reflected back on my peppermint work, where the stimulation occurred after there was an inhibition. There was a compensatory response. And so what happened is that I began to find that there were lots of examples of both, that you could have direct stimulations or overcompensation stimulations. And what really struck me was that it didn't make any difference on the size of the stimulation. It was always in this maximum 30 to 60% range. And I said, wow. I said, that told me that the hormetic dose response is really independent of mechanism, because that there are diverse mechanisms, and it's all and they're all constrained by something. And I developed this idea that it was constrained by the limits of biological plasticity. It was pushing the system to as far as the system would allow itself to be pushed, and the system, kind of like nuclear power plants and so forth, you have you might say overlapping protective systems. If this system fails, another system comes in to prevent the failure, and a third system comes in to protect that failure. And I'm saying, wow, it reminded me of that. I said, it's very hard to break through this system. I haven't really seen examples of that. And so I came away thinking that from an evolutionary point of view, somehow within nature, that nature has thought that the way to enhance survival is to have some flexibility in the system, allows yourself to compensate when you're stressed, and but that compensation is limited, and it's limited up to a certain level, and that actually has to be genetically programmed and selected for. And when you look at this from bacteria all the way to humans, it's remarkably the same. It is so striking. And the odd thing is that the biological, biomedical, and other communities that have missed this for 150 years of very intense study in all different kinds of countries and all levels. And so I got into trying to also figure out, well, how could our really very brainy, very outstanding biological and biomedical communities miss a fundamental concept, and how could this possibly happen? And if you could miss the one, well, maybe how many other concepts are people missing, and what are some of the reasons for that? And so it drove me into a lot of regions of thought, and that I had never anticipated thinking about before, becoming much more philosophical and much more historical. And because scientists are human, then they operate in some sort of a structure at a time, and their thoughts are constrained by social and other factors.
Well, the general concept. So I first, I guess, was really exposed to this in the neuroscience field from the concept of preconditioning. So actually, this, I think, it was first established in the heart, but then later in the brain. So these experiments where you, in the case of the heart, you block some blood flow to the heart through a coronary artery at a low level that doesn't damage the heart tissue, and then you wait some time thereafter, a day or whatever, and then you come back and you essentially give the amount of ischemia that would have otherwise caused a lot of damage to the heart. For having done this preconditioning ischemia, it protects the heart, and that's also been shown in stroke models in the brain. So that kind of idea, exposure to a mild, moderate, maybe moderate, I'd say, stress can have a beneficial effect. So that preconditioning concept does fall under the umbrella of hormesis, I would think.
Well, as it turns out, because in trying to take a look at the history of preconditioning, I found, to my great surprise, that somebody did a study with radiation in plants published it in 1927, where they gave a low exposure to radiation, and it protected against a higher dose of radiation, and they got a stimulation of the growth with the low dose of radiation. And then that work proceeded, actually. And then around, I think it was in the late 40s or early 1950s, a fellow from Europe by the name of Pepe, he did a much more comprehensive, I'm going to say, dose response with low dose radiation, low and high dose radiation. He covered the dose response continuum and followed with a higher dose. And what he found was the magnificent, what I call, biphasic dose response relationship. It was a hormetic dose response framed within a preconditioning component, even though the earlier work supported it all, that you knew the preconditioning would occur and so forth, but he actually extended it to a very broad dose response relationship. And so it's such a joy for me to see this hidden in a German journal that was, in the 1950s. It was published in the German language, so I had to get it translated. I mean, even though I took 2 years of scientific German when I was an undergraduate and so forth, but I got it translated by a translation service here. And because for me, it became one of these hidden gems in the scientific literature.
Yeah.
That if somebody had actually been more widely read in 1950s, early 1950s, and they had seen this, they would have seen, wow, when all the debates were taking place about radiation and how to address it and so forth, which was really contemporary with the mutation developments and so forth, they would have seen the greater complexity of the biology, put it that way, and a bit more open to biological diverse questions. And
in addition, one, I guess, last thing on evolution, maybe last thing before we move on, is that, so we've done this work with phytochemicals, chemicals produced by plants, and their effects on neurons. Others have looked at other cell types. And so it turns out many of the chemicals in the skin of fruits or the vital parts of vegetables, the things, broccoli sprouts, broccoli heads, and so on. The reason those chemicals are produced by the plants is to keep insects and other organisms, including us, from eating too much of those plants, particularly the vital parts. So they're actually natural pesticides or natural antifeedants. But during evolution, it was to our advantage of herbivores, to be able to consume these plants because they have energy, they have calories, they have other nutrients. So there's this interesting coevolution that occurred over, tens, hundreds, thousands, millions of years, where we evolved mechanisms to protect ourselves from any potentially toxic effects of these chemicals. And there's kind of four mechanisms. The first is, turns out all these chemicals have a bitter taste. They're very bitter if you take the pure chemicals. So I'm talking about sulforaphane, curcumin, caffeine. The most widely used drug in the world, caffeine. It's very bitter. If you put tea leaves or coffee grounds on your kitchen table, ants won't touch it. It's because caffeine is the reason. But anyway, so it turns out bitter taste. The second one is vomiting, right? If you get too much of these chemicals. The third mechanism is there's enzymes in the liver called cytochrome P450s that rapidly remove these chemicals, detoxify the chemicals. So they're in our system a short time. They induce stress on our cells. We've shown this, others have shown this. Activate one of the pathways, very classic stress response pathway called NRF2, ARE pathway, and it's a pathway that stimulates genes that encode antioxidant enzymes. But there are other pathways, repair pathways, DNA repair pathways, that are stimulated by these toxic chemicals. So that's kind of an interesting example of the coevolution. But one thing I want to talk about from the Hormesis standpoint, we've talked about the dose, but the duration of exposure and the frequency of exposure, I think, is very important. We found this in some of our studies. You briefly expose neurons to, well, one example is glutamate. You expose neurons to a high level of glutamate that would normally kill them if you kept them exposed to the glutamate for, say, several hours or more. But if you just expose them transiently for 5 minutes, and then you wash out the glutamate, the neurons survive. And then if you later expose them longer to the high level of glutamate, they're more resistant to being killed. So the duration of exposure to a given concentration is important. And then I think frequency, exercise is a good example, right? There is a limit to how much we can exercise without it having detrimental effects. And so I think the frequency may be important. And then the last thing I'll say here, I'm saying a lot, but I got a lot of thoughts flowing through my mind, is that medicine, drug development, it's all focused on, find a dose of a drug that has a beneficial effect, and then try to keep that dose, at the same level. But from the standpoint of activating hormetic beneficial pathways, I think the, intermittent exposure and allowing a recovery period is important. This recovery period then allows, with exercise, it allows your muscles to actually grow more mitochondria. Your muscles don't get bigger while you're exercising. They get bigger during the recovery. But if you don't exercise, they won't get bigger because they haven't had the hormetic challenge that's necessary to stimulate those evolutionary important pathways. So anyway, I've said a lot there, but I'd like your thoughts on all of this.
There are actually a bunch of things there, Mark. But one is I'd have to agree with you on the bitter taste, because I take some supplements, and I don't like taking supplements. I'd like to just put them all into a little swish of soda or water and drink it down like that. So I've tried to take capsules and dissolve the capsules and then just consume a little mixture of a couple of agents, right? And while I can tell you, I agree fully that the taste of some of these is tough to take. And so taking them with capsules so that you don't have to actually taste something is people have figured that out a long time ago before I went and tried that out, right?
Well, that's why kids don't like to eat broccoli or, some of these vegetables, then things that we know are good for us, because they have a bitter taste.
And well, it's interesting on all these different effects. I have, and it's relevant to the biomedical application, and that is that there's been so much discussion in journals and in popular papers and magazines on lifespan, and can you increase lifespan? And lifespan is a very integrative endpoint. It's a biological endpoint. And so I said, well, in fact, it was interesting. There's a fellow in Europe about 20 years, probably 20 years ago now, maybe, was offering a large amount of money to somebody who could prove to him that humans couldn't live 1,500 years. And so that all these biogerontologists, he's offered this challenge, and he has this big chunk of money, over $1 million, whatever it was back in those days. And he said, and none of them could discredit what he had to say. And so I said, well, I could discredit what he had to say. I can get $1 million from this guy. So I sent him a big box of duplicate copies of everything that I had on Hormesis, wrote up this big argument for it, sent it over. I said, if this is real, I'm going to get $1 million check in the mail pretty soon. And so as it turns out, he was more or less dismissive of what I had to say, without without a substantive response. But I remembered the issue of longevity, right? And relating it back to Hormesis. Well, in the last 20 years, what's really happened is that this little nematode worm has taken over the experimental domains. It's called C. elegans, and it's well characterized genetically. It has all these really unique mutants that allow you to break down and study pathways of activation and so forth that relate to handling stress. Stress could be anything, oxidative stress, or could be gene stress from an Alzheimer's related gene stress. It could be any, or longevity. And I said, well, if I look at I'm going to look at this literature. And I found that there were probably, I'm going to say roughly 300 agents that had been tested to and published in the literature, looking at the effects of these agents on longevity in this nematode model. And almost all these chemicals were able to extend the lifespan of the nematode. And that's probably why the data were published. They have positive findings. And the interesting thing was when they did a complete dose response so that they could look at low to high doses, they showed this beautiful biphasic dose response in almost every case. And when they didn't do enough doses, they found that, yeah, they got a dose stimulation, but it was only, you know, in this modest 20, 30 or so % range. But yet with these models, they were actually able to figure out what pathways were determining. And you're one of the co-authors on these papers, Mark, for sure.
Yeah, on one or two of them.
Yeah, because I remember reading
Yeah.
your papers, and so I came to the conclusion that actually what was really the overall determining factor for lifespan extension was actually hormesis. And it actually didn't make any difference whether, and there were probably 5 pathways or 6 pathways that were really instrumental. Sometimes the pathways talk to each other, sometimes they don't seem to talk to each other. But it really didn't make any difference whether they were given a single chemical, like a pharmacological chemical, well-characterized chemical, or they were given a mixture. There was an extract from a leaf of a plant that had 1,000 different chemicals in it, 10,000 chemicals in it. If it stimulated the capacity for these pathways to ultimately lead to lifespan extension, they were all limited by what appeared to me the bounds of biological plasticity, which were described by the quantitative features of the hormetic dose response.
Yeah.
And I said, wow. I said, what it really means is that yes, life can be extended, but it only within narrow limits.
Yeah. Yeah.
And when I look at people living today, I'd say that people are definitely tapping into their hormetic potential, because they may live mostly a vegetarian life. They may be exercising. They may be handling other kinds of stressful ways that are positive.
Low calorie intake or intermittent fasting type.
That's right.
Yep.
And so I think they're tapping into it. And so when somebody makes it to 90, 95, they have really, if you want to take a look, I don't know, what the baseline would be, but it seemed to me that it's, and this fellow who was thinking 1,500 years, in fact, I thought that people could live out to 150 to 180 years, but I think it's mostly, around 110 or so, or might be the maximum. And we're really tapping into a lot of hormetic potential, but not all. And we don't know, how early it really needs to be tapped into to affect, later on in life and so forth. But I think that this is very significant development, and it's something that I just had a paper that was published in one of the journals on making this claim for hormesis and longevity. And then a paper currently is under review now, actually, in which I have all the backup data to make the claim. And I'm fully expecting it'll be well received and it'll make it through the system. You never know what reviewers will think of what you write. But yeah, but it taps into. But the other interesting thing here, Mark, was that in this nematode model, it was, if the animals were healthy, you could go right out and you get your lifespan extension, but also that the same chemical is also helping these nematodes that have cope with stress, other stresses. So that if somebody's, if you could live, so if you're just a normal healthy nematode, you get your 30%. But if you happen to have, and they tie in, an Alzheimer's gene, they tie in the equivalent of a stroke, or however model they want to frame it within this system, you can get protection, up to
against multiple types of stressors that are,
yeah,
it could be an oxidative stress or a metabolic stress or something we call proteotoxic stress or accumulation.
right.
I want to say one thing about the worms. So, so this, what was it, $1 million I can get?
Yeah, back in those days, yeah.
Yeah, yeah. But in any organism.
Well, the more
So
or less,
these worms, there's this condition, if you deprive them of food, so normally you grow them on this, they eat bacteria.
right, yes.
So if you deprive them of bacteria, they'll go into this, it's kind of a hibernation,
Diapause, yeah,
suspended animation mode where
right.
they can stay alive, but they're not really functioning. They're just sitting there doing nothing for a long time, like 10, 20, 30-fold their normal lifespan. And then there's these other organisms, this all fascinating stuff. They're called tardigrades. You know that?
That I don't know.
There's these organisms, what are they called? Water bears or something? They, if like they're live in these little streams or by them, and if it goes dry, there's a drought, then they desiccate, they completely dry out, but they're not dead. They can, when it rains and they get hydrated again, they come back to life. So there's anyway, I guess I'm just thinking, to me that doesn't really count so much as extending lifespan, because you're just interrupting lifespan.
Yeah, you're taking them out, putting them on the side. 'Cause when I was going through my PhD program, I worked on a similar kind of a thing with insects, but it was called diapause.
Yeah.
And so yeah, but I
Actually, Ed, I worked on molting in crabs and the neuroendocrine system that controls molting for my PhD work. So I know about insects because they also have the molting hormone, ecdysone, and
that's right.
yeah.
Yeah, that brings me way back.
Yeah.
Oh yeah, that's for sure. But I wished that I wished I knew then what I know now. I would have done, a whole lot of different sets of experiments
Yeah.
than we
I,
did, even though,
I
yeah,
want to say on, your point about, we can maximize our lifespan. We know a number of hormetic challenges that can maximize our lifespan. Regular exercise, moderation in energy intake, probably intermittent fasting. From the standpoint of brain aging, keeping intellectually engaged, keeping your neurons challenged. Right now, your neurons and mine and our hippocampus and related brain regions, they're very active right now because we're trying to think about what we're talking about, trying to remember what you talked about and so on. So just like when your muscles are active, when your brain cells are active, there's increased oxidative stress, big elevation of intracellular calcium, which can be toxic if it stays too high, and metabolic increased ATP utilization. But it's good for them as long as we sleep and get rest, so that recovery period again. So the big problem in the United States and industrialized countries is people are not subjecting themselves to these intermittent hormetic challenges.
well,
The typical couch potato stereotype is shortening lifespan, or at least, leading to submaximal lifespan.
well, it's interesting, Mark. I found that because healthcare is very expensive. I'm within a school of public health, and people try to say, well, how can you maximally benefit the entire population, but especially those that might be economically compromised and have certain greater challenges, put it that way. And I've said a lot through writings and presentations that actually you don't need a lot of money. You just need some knowledge, and the knowledge is tied into, people might say, well, it's really because I eat one meal a day. I mean, everything I do is designed to stress myself. I mean, I will exercise later. I will, I'll take a cold shower for a certain number of minutes. I will do many different things in the course of a day that are designed to actually stress different aspects of my system, and I think that people can as implied in our conversation, there are so many ways that by eating less, eating wisely, but eating less, and knowing how to pick and choose things. Knowledge is our greatest asset here. And one of the things that I've found on my Hormesis quest over the years, and about 3 years ago, 2 or 3 years ago, I published 14 papers on 14 different stem cells, all on Hormesis, from bone marrow to epidermal to you name it, all the whole range of stem cells, adipose stem cells. And what I found was that every single stem cell, the literature was just overwhelmed with hormetic examples. And I found that in almost every single case that the term hormesis was never used.
Yeah.
And yet it was there. So and so I'm saying, well, I'm not a stem cell person, but I'm looking at this literature, and I see in that literature hormesis is actually pretty common. So I write 14 papers, try to send them to good journals, get them all published, and then realize that the people who are doing this research don't know the concept at all. And so then what I started to do was to then have after the introduction of the paper, to then have a little hormesis overview. You're going to be seeing something that you've seen these sorts of things before, but you don't have a broader context of how to frame them. And I think that one of the challenges in this world of biology, biomedical research is that we're all so highly specialized that it's hard to know what's going on outside your field. And it's quite interesting, Mark. It was really due to back in the late 1990s, maybe early 2000s, that the field of gerontology was maybe the first to really embrace the hormesis concept with language. They used the hormesis language, and it was really tied into about 4 or 5 real leaders in the field. I mean, you being one of them. There was people at the University of Colorado, doing.
That was in C. elegans too.
Yeah, the CLAs. Then there was also the creation of the Biogerontology Journal with Suresh Ratan. There was about 3 or 4 individuals, and they used the term hormesis. And
we had, I was the founding editor for the journal Aging Research Reviews, and over the years we had multiple articles.
And so because of those two journals and the leadership at the very high, I had this I read a very interesting, it was kind of an I don't know, anthropological sort of a study. But these researchers were looking at 2 groups of baboons, and they had their social structure really well characterized. And in one group, they taught the top male how to gather food when it was placed on the water, a certain sort of food, and it was very important for feeding and energy and whatever they would need for survival. And then they taught when they taught this group, everybody, all the other baboons copied it within about 15 minutes. It was very, very fast distribution of knowledge, right? Then the other group, they taught it to the lowliest female baboon, the exact same information, and that the low one on the totem pole, so to speak, and it took 4 or 5 months for the group to ultimately copy and to do this. And so
So they pay attention to the leader.
yeah, that's right. And I said, well, the reason why it was interesting that Hormesis made it within the biogerontology community is that, 3 or 4 strong leaders adopted the concept language, and then other people picked it up. But in other fields, it's the concept may be there, but it's very difficult for it to filter up. And how this works.
So humans, following the leader, it could be good if you have a good leader, but if you have a bad leader, it can cause a lot of trouble, as we saw with the previous president.
Well, that the interesting thing too, Mark, along this line is that it also shows, how siloed the scientific disciplines are, because biogerontology is such a major field. It's really a major field. It's central problem that people are trying to wrestle with, and it's major advances. And you have, well, I mean, are stem cells relevant to lifespan? Yes, they are. And these people have to be, cross-discipline communication. And I find that one of the big problems with that probably any new concept has, and that is, well, how do you break through that knowledge to other disciplines so they could benefit from it, and then synergize with your own? And I think that's a serious issue that
it definitely is. You and I, some scientists have a lot of tunnel vision, right? And they ask this one highly specific question. They write a grant proposal on that. They stick to the specific aims, and then get their grant renewed, and to elaborate a little on that specific. And they're probably not reading, anything much at all outside of their specific area. I think that can be a problem. So the take-home message there is, I used to go to the cell biology meetings, and I kept doing that even when I was a neuroscientist, and I'd often learn a lot at the cell biology meetings, some of the latest findings that people in the neuroscience field didn't know about, because they're, so focused on the neuroscience. Then I'd take that knowledge that I gained in that, different subfield and apply there. And I've done that over and over. I think it's been really useful approach.
Right. Well, it's interesting, Mark, because when you have a concept like hormesis, what makes it really significant is the statement that you really introduced the talk with, and it was really evolutionary biology, and how it really comes to bear within that, and that is that you really have to see, you have to really take a big broad look at life, and within the hormetic thing, well, gee, you're really seeing this hormetic response. I'm seeing it, in all from, as I said, bacteria up to humans, in plants, and all different cell types, and the low, and all over. And yet, that in our very specialized world, that people only look at something that they know about. And my concern with hormesis that I have is that I feel that if I can have 3 hours of your time and you could sit down with me, I can tell you why I believe this is true and what its applications are. Can you give me 3 hours of your time? And it'll change your life. It'll change the way you think. It'll change not only your profession, it'll change how you live your life, actually, and your habits. But I need 3 hours of your time. And so this is one of the real challenges of public education. And it's also, especially when you're predisposed to not being open to ideas. And I think,
and when there's forces, profit-driven forces pushing against it, big agriculture, monoculture, processed food industry, fast food, even pharmaceutical industry. Pharmaceutical industry, there's no work on, really hormesis, or the notion that can we develop interventions that engage some of these evolutionary conserved pathways rather than wait till somebody gets sick, with a disease and then try to treat it once they already have it. So the prevention, the risk reduction prevention, there's no money, there's no, all these profit-driven forces are in a way pushing against that, because they're not going to make money if people have healthy habits and don't get sick.
Well, I come into this world from an environmental perspective, and hormesis, and there's a very interesting intersection, and it relates to your work on dietary restriction, and that was this in the world that I live in, where they're testing chemicals for toxic effects or cancer, things of that nature. They feed the animals ad libitum, so they let them eat as much as they want over the course of their life, and the rats come out very very overweight. They're not exercised. They come out like little blimps after 2 years, and it doesn't resemble actually what goes on in a human life. And there was a fellow by the name of Ron Hart, who was the
I know him.
He used to be the director of the National Center for NCTR, toxicological research for the FDA down in Arkansas, and Ron was very taken with now what happens if you reduce the caloric intake from this ad libitum? How would it affect toxic responses? Well, this was in the late 80s, early 90s, and what he found was, my God, these chemicals that were so toxic, when you fed them, when you allowed ad libitum and you had the animals eat all these calories, get very fat, which is probably also an incredibly strong, inflammatory stimulus. And so what he found was that it actually greatly reduced the occurrence of adverse effects. And so he had, wow, you could really change public health, but then how do you study these chemicals? What's the best way to do this? And I linked up with him because he started looking at different degrees of caloric restriction, and he found these beautiful inverted U-shaped dose responses. And so we kind of converged on, wow, along these same lines. And there were some other early people who were doing this. Ed Massaro, who was at the University of Texas down in San Antonio. And so when I was every year doing my hormesis conferences, I would bring these and other people up and try to get these concepts together. But then ultimately, it led to a lot of your work on intermittent fasting as a means of stressing the system to upregulate protective mechanisms, and how all this fits into lifestyle, and how it also fits into the challenges to groups like I'm going to say the FDA or the EPA. How do you do your studies? How do you estimate, if you're going to be estimating human risk from animal studies? Do you do this with animals that are being fed ad libitum, or do you do this with a range of sorts of diets? And how do you fit this into a whole testing scheme? And I don't think that they've really probably had the opportunity to think about it, but there should be some revolutionary thinking that should be taking place within these organizations. And
I had the same exact conclusion about, and for me it started. So I was any animal studies you do, any experiments you propose to do, you have to run what's called through the Animal Care and Use Committee to, make sure you're treating the animals okay and they're not suffering and so on. And then, so we're doing these. This is when I first got to the National Institute on Aging, the very first year. And, you know, they're proposing to do these intermittent fasting and caloric restriction studies, and I put the proposal in, and they said, well, you need to justify, you know, how this is not having adverse effect on the animals. And I turned it around. I said, everyone else need to justify why they're feeding the animals ad libitum and they're not getting any exercise, because that's the unhealthy condition. But then I with a couple postdocs, I wrote an article for the for PNAS, and the title of it was "Laboratory Rodents Are Metabolically Morbid: Why It Matters." And it's the exact same thing you point out there, that it makes a difference in a lot of different things you're testing, what that, I guess, baseline hormetic level of hormetic system engagement is.
Well,
Right.
you know, yeah, it's interesting along those lines, because you have animals that have really such you know, it's morbid metabolic state. I mean, it's a very highly inflammatory condition.
Yeah.
One of the things that cycle back within that framework, and that is that, you know, I've looked at inducing hormesis over time, over the lifespan, and 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 got into this in certain interesting ways, that as you know, at least in rodents anyways, you could see hormetic interventions being very successful in young animals and those that are reaching middle age, put it that way. And then when you reach middle aged, and then you start to get older, say the elderly rodent, I'm going to say 24, yeah, it goes downhill, and and uh, and you're losing this capacity for this 30 to 60%. It's it's interesting that what I'm in the literature, based upon I'm going to say a variety of models, that as you age, what I found was that the maximum stimulation actually goes down, and the width of the stimulatory range constricts. This is like a new concept. This is,
Oh,
to me, this is all within the last 6 months or so, based upon looking at the C. elegans and other sorts of things. But I found that some aspects that are hopeful, and that is that some investigators, not a lot, but some, after many failures, you know, working with elderly mice and rats, put it that way, that certain, depends on the age, and depends on the gender, and depends on the strain. You might think some of these things are, you know, they're very specific, but with certain exercise interventions and certain dietary interventions, and their combination, that you can, you might say, take a pathway that had been degraded, so to speak, and to restore that pathway, and then you can restore some metabolic, you know, some hormetic function to that, you know, I'm going to say 24 to 30-month-old you know, mouse or rat.
wow.
And
Yeah.
yeah, it's actually, you know, pretty amazing things. And so it shows it really shows flexibility within the system, and able to reconstitute that. And to me, that's a very significant possibility here. And so I'd throw that out for discussion.
Yeah, absolutely. It's never too late to get some benefit from hormetic challenges. I think you know, this earlier this year, we got a sauna, infrared sauna. So I'm doing that a couple days a week, you know, and get drenched in sweat. There's some pretty good data on that.
That's right.
But returning to the brain just briefly, I wanted to mention that this hormesis concept applies to some interventions that have been used by neurologists in the past. Perhaps the most profound example is electroconvulsive shock therapy for people with severe depression that doesn't respond to, antidepressant drugs or the psychotherapy. And just one or a few electroconvulsive shock where they essentially induce mild seizures, which is, it's obviously stress. Again, this gets back to glutamate. It's essentially causing increased activity in these neurons that use glutamate as a transmitter. So that intervention, a transient kind of, and this is quite a bit of stress actually, but for a short time period, can have a long-lasting beneficial clinical effect in relieving depression. And then I think the psychedelics, right, which is, I don't know if have you looked at that yet? I can't remember.
Not really.
There's another paper probably. I don't know if there's enough data on dose response, but there's a lot of work going on now with psilocybin, the chemical in magic mushrooms.
Right.
And really good data on relieving depression. Some things coming out now on addiction, either to alcohol or drugs, the psychedelics. So again, it's like a transient exposure to something that's causing a profound effect in the neural circuits. And it must be some kind of stress, you have hallucinations and so on. Has a long-lasting beneficial effect. And then we've explored this idea. We did all this work on intermittent fasting with subject rats or mice to intermittent fasting for several weeks or months, and then we exposed them to levels of neurotoxins that would normally damage or even kill neurons. So excitotoxins such as canic acid or domoic acid, which come from algae, or there's this, and I've had previous episodes on this toxin, MPTP. It was discovered actually as a contaminant of heroin. And how's your time? Did you have something else?
No, I have
Okay.
my time is totally free.
Okay. Yeah, anyway, that's an interesting story. A neurologist out in California, they had these patients coming in. They were young adults, like 30 years old, for example, coming into the clinic with symptoms indistinguishable from Parkinson's. And they developed these symptoms rapidly, just over a period of a few days. And it turns out it was through this real good investigation, kind of a detective work, they found that these people, that they came into multiple hospitals actually, they were drug users, heroin users, and they had all used the same batch of synthetic heroin. And the chemist who made the heroin messed up, and there's this other chemical, MPTP, in it. And turns out it's a selective toxin to dopamine neurons who use dopamine, and those are the neurons that degenerate in Parkinson's. But anyway, we found this intermittent fasting pre-treatment would protect dopaminergic neurons in mice against degeneration. So again, we have an example of a physiologically relevant condition, fasting, protecting against a toxin. It's pretty remarkable. But anyway, we've tried to mimic the effects of caloric restriction or intermittent fasting or other stressors pharmacologically. And this started in the 90s, actually, where I had a couple of postdocs, one who was an expert on a stroke model, another with this MPTP Parkinson's model. And I had them administer 2-deoxyglucose once daily to these animals for a number of days, and then subject them to the stroke or the MPTP. And so 2-deoxyglucose, it kind of tricks the cells into experiencing caloric restriction. It causes caloric restriction at the cellular level because it won't let glucose be used by the cells, and actually it will cause ketosis, just like fasting. So that's kind of interesting that 2DG once daily. But then we did a lifespan study at the National Institute on Aging. A colleague of mine, Don Ingram, did that study, putting the 2-deoxyglucose in the food. And in this case, we fed them ad libitum. So actually, they're having like continuously 2-deoxyglucose at a lower level actually than the single dose we gave the animals in their study. But it turns out it actually didn't extend lifespan. It shortened lifespan. It had bad effects on the heart.
Really?
So I'm thinking there, you're continuously, you can't subject yourself continuously to calorie restriction, right? You'd starve to death. So this intermittent challenge seems to be really good for neuroprotection. And then finally, more recently, we used another, it's actually a toxin you probably know of, 2,4-dinitrophenol.
Yes, I do.
DMP.
That's right.
And this is used by chemists and people in the lab for certain chemical reactions and things. And it is toxic at high levels, no doubt. And what it does is it causes what's called uncoupling of oxidative phosphorylation in the mitochondria. So the cells can't produce ATP as well as they normally do because you have this, essentially it's causing a proton leak across the membrane. But anyway, we found that low doses of DNP are neuroprotective. And there's this company actually that's trying to get low dose DNP into the clinic for certain disorders. So I think those are some kind of examples of translational work that's kind of interesting. Low dose toxic things, 2-deoxyglucose, DNP, can those be used clinically, administered in an intermittent manner, not in a continuous manner?
You know what I found it was interesting is on this aging research, Mark, and that it was fascinating that let's say there are 300 different agents that extended the life of the nematodes, right? And they all kind of extended the nematodes to roughly the same degree. There's some variability within that, but I'll say 15 to 35% in that sort of a range, right? And there really wasn't one chemical. You say, well, what's the best one? Well, actually, they're all kind of about the same. In a sense. I mean, you can, and people are testing all in the commercial interest. They're testing all different sorts of agents and permutations of agents, and many of them they act on. They're going to act on one of the 6 pathways, or maybe most of the 6 pathways that are identified, and they all come out with about the same answer. And so I might say, well, okay, well, and my perspective is that, well, okay, I'm going to get what happens when you have a mixture of these things, right? So can you get synergies, and can you have hormetic synergies, and can you, if you take 6 of these hormetic acting agents that extend lifespan, and you put them in their optimal concentrations, can you go from 30% increase up to 300% increase, or 100, whatever? And actually, as it turns out, I believe that you can't. I believe they're constrained by the limits of plasticity, because some of these extracts you get from, it depends on the plant, but you might have 5 or 6 really really good hormetic agents within that extract. But when you put them all together, they don't do any better than just one. And
Well, and this goes back to what you said about humans, right? That yes, we can extend our lifespan by moderation, subjecting ourselves to relatively low energy intake, regular exercise, keep your mind intellectually challenged. But, no matter what we do, we're probably, I kind of agree, the systems are all geared for not having these dramatically synergistic effects. And in the lifespan studies, the exercise actually didn't synergize with caloric restriction. In other words, if you put running wheels in the cages of animals from when they're young and caloric restriction, they don't live really much longer than just caloric restriction alone.
Along those same lines, there was some really interesting work showing, looking at chemicals that could increase memory within mice in different studies. And many of these, and there are actually several, dozens and dozens of these agents that were successful, and they had nice dose responses. So all showed these inverted U hormetic dose responses, all within this 30 to 60% range for increases for the most part at the maximum. So when they combine them in a nice dose response metric, so they had all kinds of dose combinations of up to 3 agents tested at once. They had synergies, but they synergized at lower concentrations, and they maxed out at this 30 to 60%. And so you could have got to that same improvement with one agent at a certain dose, or 3, but in some sort of a lower combination of doses for those 3. But they were all, so this was leading me again. It all came back to, wow, this 30 to 60% rule. It's like a biological rule, and so I think there, in terms of hormesis, and since I'm 77 years old now, okay, and I'm saying, well, and I'm looking at this data where the hormetic benefits dropping off as you get older, and I'm saying, well, I mean, and I'm exercising, and I'm doing caloric restriction, I'm intermittent fasting, and I'm doing this and I'm doing that, and I'm saying, well, to what extent am I at this age activating? Am I activating anything? And am I activating it the right way?
And I'm sure you are, particularly. Yeah, I'm sure you are.
But I thought back to some. I have different. We all know, and probably ourselves. But different people have different diseases that we know or have experienced. And I mean, I've had 3 friends with ALS. One passed away, and others with Parkinson's condition, and others with Alzheimer's and so forth, and so for these, especially for my ALS, the several, I spent a lot of time in the literature and looking at, are there ways in which hormesis could be helpful to slow down the onset of some of these symptoms for any of these conditions, because so I did a massive amount of work, I can assure you, to try to come up with something to save my friend's life, and its quality of his life, right? And I think that this is, how does hormesis relate to that? And I've thought along these lines, and I said, wow, now this may not be right, but I think once the disease starts is clinical, and it's really identified by the physicians, and they're quite sure that this is it, that disease has probably been developing for many years, and it's like a snowball rolling down like a mountain, and by the time they identify it, it's got a lot of momentum, and you put up a little fence, it's going to roll over that fence. I think that it made me think that hormetic interventions for conditions that are likely to develop anyways, that you need to develop a hormetic lifestyle early.
I agree.
And that can these disease process, even in the nematodes, when they insert the Alzheimer's gene or Parkinson's predisposition or something like that, they'll still get it, but the intervention can push it down the road,
Yeah.
if you do it early enough. And so my sense of most people, they life is great, you're 20, 30, 40, and when you start to get serious about your health, maybe your diet at 55 or something like that, you'd like to intervene, because people, when they're younger, they think they're going to live forever.
Yeah.
And they're going to, life is okay. But this is a time when you actually can, you are aging and you don't know it.
Yeah.
And so these,
I'm getting ready. I teach a course on the neurobiology of aging for the graduate program at neuroscience program at Hopkins. And, I'm getting my lectures ready. And the first lecture is number of slides, showing age on the x-axis.
Yeah.
And some neurological function measurement on the y-axis. Everything started pretty much, most things start declining actually when you're in your 30s. Things like even reaction time, certain variables of cognitive function even. And this is something I think we all notice, like it comes up again historically. People tend to be most creative and do the best work when they're in their young 30s and 40s, for example. It's true with musicians. I grew up in the 70s, essentially, 60s and 70s. And I look back at those people, performers, all their good work, their best albums were their early work. Even when they get in their 40s, 50s, they decline. So, yeah, you're right. People need to think about this from the time they're, say, 20. They need to think about aging.
Well, and there are so many simple things that one can do to enhance your capacity for protect what, and it's interesting, Mark, because I do a lot of things to try to protect my biology, put it that way. A lot of things, a lot of different low-level stressors, put it that way. And so some friends will say to me, well, how do you feel you're taking this? I said, well, I don't really feel, you see advertisements on television say, oh, I have energy, I have boost, because I take the product. It's an advertising product. I said, I'm taking a lot more than what they do. I know kind of what I'm trying to do. I've seen a lot of the data behind it. And I can tell you that every intervention that I have, it doesn't make me feel better. I mean, I said, it really probably shouldn't. I said, what it's really doing is slowing down protein oxidation. I said, it's recycling damaged protein and cleaning things up. It's slowing down my rate of aging. It's
well, the one thing that definitely makes people feel better is exercise. It has a huge effect, right? Exercise. So, I've talked to you about this before. When I had my mountain bike accident and had the multiple surgeries and stuff, and couldn't exercise for a number of months, my mood went way down. I got depressed. Then when finally I got able back to start getting some meaningful exercise, particularly aerobic exercise, then everything turned around.
Well, I'd have to agree with you, Mark. The one thing I probably haven't shared with you, and that is that we have another thing in common. You had the mountain bike accident, which was pretty rough. I had a road bike accident where I broke my pelvis in 2 places, and
wow.
I did that at age 65, and I'd finished a 50-mile bike ride. I was 1/2 mile from home. I don't know what happened. I looked at my watch. It was a Sunday afternoon. I was there. Oh, good. It's about 2 in the afternoon. I think I'll watch the Patriots in the second half of a football game, right? and within like, you know, a millisecond after that, I got unstable on the bike and went down, and I fell down, thank goodness, right next to one of the fire stations in Amherst. So they had
But did you hit on the bar of the bike with, how did you break your pelvis?
I landed on the road.
On the road. Okay.
Yeah, I landed on the road, and how I fell must have been very awkward.
Yeah.
And broke it in two places, and I have to say, you know, then I was fortunate because I didn't need surgery. It somehow there were two breaks, but they were aligned properly, and I was able to recover, you know, more effectively, put it that way. But I was out of action for a while, and I mean, I can understand how if you were like older and not in shape, and it's very hard to get out of bed because the pain is pretty rough.
Yeah.
Pretty rough. But you know, but the thing of it is you know, recovery is an exercise. Ultimately, you know, pulls you through
Yeah.
a lot of that. Even I think that if you have been doing these hormetic lifestyle features and you do get hurt, I think you recover better.
Yeah.
More quickly, and things that in terms of my work anyways, I've tried to take a look at a wide variety of things, areas that I've known nothing about, but that I felt were really important, maybe, and I could learn something as kind of a carpetbagger going into, well, the area of not just, you know, I'm going to put it this way, certainly the stem cell area, but all these stem cells, but the area of wound healing, and how quickly, you know, could we accelerate how fast bones could heal, or our Achilles tendons, or, you know, or just a regular wound, or how do you do something for somebody with diabetes?
Or surgery, recovery from surgery.
Yeah, and the interesting thing is it wasn't really much different than these you know, and than the stem cell area. There was an awful lot of excellent work, you know, showing enhanced protection, showing hormetic dose responses, you know, looking at the key cells and the overall process, you know, whether it's fibroblasts or keratinocytes, or tying them all together in the process. And you saw actually the dose optimality. You saw the beautiful hormetic response. You get a stimulation up to about 30 to 60%. It's inherent in like every system, you know, that we have, even you know, protecting I'm going to say, you know, when you're looking at, I have a friend whose wife has glaucoma and macular degeneration. She has both of these things at the same time. How can you slow down the rate of either of these things, right? And it's interesting when you go back and then you look at the studies that are underlying research in this area, where you might have some capacity to slow down these disease processes. They almost always reflect a hormetic dose response.
Yeah.
And the investigators have no clue of what they're working on, and why or how this is conforming to a particular, you know, dose response function. They are so into their, and it's good that they're really into their narrow world, because we have progress there, but it results in them not understanding the limits within which they're working, or the opportunities within which they're working. You know, that sort of thing. Is there's a you know, one of the major challenges in the hormetic area is simply to educate. And you know, problem with hormesis is that it was kind of like my advisor way back in 1966 identified, and that is, okay, well, you have actually compared to toxicologists are used to be kill them and count them sorts of people, and you give high doses and you see biological effects that you can publish. It's very reproducible, and you identify a toxic effect. It's kind of a very simple sort of a descriptive sort of a thing. But when you really get down to where we live in the human world, the exposures are much less. You have diseases that are progressive, and they're not, you know, just jumping out at you. and you begin to see that biology is really played out within the bounds of plasticity.
Yeah.
And that's where hormesis is found, and that's within the evolutionary strategy. And this is such a big it's a way of, we really have to re-educate our biomedical
Yeah, and until that happens, there won't be a critical mass of scientists and even clinicians that are pushing government, policymakers to change things in a way that's gonna.
Yeah, it's from the textbook. It's so how do you change? It's like changing the direction of an aircraft carrier. It's got so much momentum in a certain direction, but it needs to change direction.
Yeah.
And how do you? And you're like this little little thing trying to push an aircraft carrier, but the thing of it is correct ideas don't die. They are there to be discovered and to be applied and to be used. And I'm so thankful that even though I started early on with hormesis, I'd have to say my approach has been I like the lowly baboon, whereas all my whatever I did, it never could have happened if it hadn't been for people such as yourself that were extremely very successful in your own in your discipline, at the top of your discipline, that others would listen to you and take your message, because they certainly wouldn't take my message, but they could take the message from leaders who were really at the top of their fields.
Yeah,
very interesting.
Well, I think we maybe wrap up now. We came back from evolution to policy and changing the healthcare system.
Yeah.
And how can better understanding of the prevalence and almost universality of hormesis in responses of biological systems to pretty much in any environmental challenge, whether it's a challenge coming from outside or inside.
Well, Mark, it's interesting. People that are real societal leaders who can advocate hormesis and do it well, and you're really one of them for sure. There are people like, for example, David Sinclair, who David gets. He travels a lot. He's a really good spokesman. He's got a wonderfully engaging personality. He goes on a lot of podcasts and he tells an integrative longevity story that's really based upon hormesis. Or was it Rhonda Patrick?
Yeah.
With her is a
I'm gonna be on with her again. I have the new book I wrote, Sculptor and Destroyer.
Oh, that's great.
Tales of Glutamate, the brain's most important transmitter. That came out last month, I think.
I'll have to pick that up, Mark. Thank you very much for telling me that.
But she's going to have me. So, but does David talk? Does he use the word hormesis?
Yeah, he does.
Okay.
I've listened to about 10 of his podcasts. They're usually an hour and a half long, and he also has like a little bit of profit-driven motive. Well, he's a complex sort of a scientist, but
he's about these sirtuin,
right?
These chemicals that activate sirtuins. He's got, I think, a whole company around that.
And Rhonda is very good at what she does.
She's much better than me than doing podcasts, because she takes the time to, get the journal articles and well, there's YouTube like podcasts where you have video and audio, But then there's just the audio podcast. I essentially, I don't put up any visuals because I think most of the people that actually listen to my podcast are listening through just listening, because then they can, and I do this, I can be doing other things while I'm listening.
So that's true.
Whereas if you're watching, and then one thing I found out from my daughter, which I didn't know because I'm not up on the technology, is that a lot of reason these young kids, the younger generation, just listens to podcasts and never watches them is because on your phone, if you go on YouTube and you have the podcast on, you have to have the video and your battery goes down really quickly.
Right.
Whereas if you just have the audio,
So why
it does, it goes, you don't use up much of your battery. So it's kind of a practical thing.
Although that's that for sure they'll go that way. Absolutely. Right.
Yeah.
So that's very, but I think that trying to find a way to reach people in influential positions, or they're really editors. For example, I'm not
good at that, Ed, though, that, I'm not like a someone who's kind of an entrepreneur.
Yeah, I'm not either. I tend to be more, I'm more focused on it's papers and research and papers and research. And that's kind of what my life is. It's not traveling around and broadcasting so much and at all. It's a very that you tend to do what you who you are, and so that tends to be what I am, and so I'm comfortable in that domain.
But this, what we're doing now, right, on Zoom, these things are, anyone in the world can access them. So I think that's, compared to when when we were going through our early scientific career, there's none of this.
No, it's good.
The only way you could communicate was actually physically go to a meeting, but then it's just the scientists. And I often wonder why there aren't more scientific meetings done electronically, because it saves people a lot of time, travel, and money.
Well, it is interesting, Mark. Hey,
do you want to do maybe we could do a workshop? I did this once a long time ago, video only workshop.
That would be a great idea.
Where it's all remote. People don't have to travel. I never liked traveling. I'd go to Europe or Japan for meetings, and it was just a lot of downtime.
It really is. Hurts your productivity too.
Yeah.
Ways. Yeah. I mean, it's too many. A trip to Europe. I mean, it's 2 hours drive for me to Boston, then spend 2 hours, in the airport waiting to get the plane, then 7 or 8 hours over there.
Then you get there and you're jet lagged for a couple days. And
and even coming back home, I mean, I'm waking up, like in Paris it'd be 1 o'clock Boston time to take a flight back to the States because it's 6 hours later, and you arrive about 6 o'clock at night their time. I mean, there's such a tiring component to the. I mean, once you're there, then I like the visiting, but it's
the thing with me, I was doing a lot of this travel when the kids are growing up and so on, and then psychologically I felt kind of guilty no matter where I was.
I understand.
Because when you're a young investigator trying to get established, it's important to go to the meetings.
Right.
Right. And so then when I'm at home and not going to the meeting, I say, geez, I should be going to this meeting. It'll help my career and so on. But then when I get to the meeting, I feel guilty that I'm not home with the family.
Well, it's that's when you have the energy and you also have the young kids, and we both went through it. I mean, I had 2 sons, and you're trying to make your career, and you know, it's striking that right balance.
Yeah.
And the right balance is and it's hard to know what the right balance is. And it's a and how to choose things, and what to volunteer for, and how do
Yeah.
you make it work, and trying to get on certain influential committees, and so you know, we all have had different strategies. We're probably successful in our own ways, you know, because we're here talking today after so
Yeah.
many years. But yeah, and then we probably somehow found something close to okay with our families, you know, and kids came out all right, and they're
Yeah.
doing okay,
Yeah.
and maybe we were in the end maybe decent role models for the kids, and you know, hopefully without you know, found some sort of balance
Yeah.
that kind of allowed us to be healthy, to grow, and to nurture, and that's and
So
not all
now,
do it.
now it's less stressful now, though, right? So you have grandkids, I assume?
Yeah, I have one. Actually, my two sons, believe it or not, live in Rockville, Maryland. Uh,
Oh,
yeah, one's a chemist who works for the NIH, and
yeah.
the other is a lawyer for the federal government. So they both get both they live like a mile apart by accident. But
Yeah, I may know where your one son works at the chemist at uh yeah, they have this center there. They develop like high throughput screens.
yeah, he works in that area. He's in
Yeah,
the
yeah, I know that place. We used that facility once.
yeah, because that's exactly his group. It's the
Yeah.
new the new group that
Yeah, they have hundreds of thousands of chemicals,
yeah,
and
exactly.
it's all automated.
That's exactly what he's doing.
And they can, they have these, like, I don't know, 96 well plates, and you can, the machine essentially can add chemical to a given dose, dose response, and then they develop really neat cutting edge ways to read out some biological process usually using fluorescent probes.
Yeah.
And yeah, we used that. I can't remember the exact assay we were trying to develop, but we used them and did develop a bioassay.
I find that it's so interesting because that's exactly the group he's
Yeah,
hired. He's been working with them now for probably 2.5 years.
yeah,
You know,
good.
within that group. I mean, he went from like a PhD in organic synthesis, and then he took a postdoc at NCI, and he worked with and got to
Well, he's in a good place because it's kind of it's a secure job.
yeah,
It's not like a company where they could
right. No, that's right.
go out of business or quit
Yeah,
working on that project. And
yeah, it's interesting. He just came back. I saw he was up here for a wedding this past weekend for his old high school friend, but he also had a conference in Philadelphia and met with a former roommate who is in industry, pretty big time industry, and they're developing their own version of this sort of procedures, and it was interesting to see how advanced the NIH programs are. You
yeah,
know, that's extremely advanced,
yeah.
and ultimately that will feed back out into, I'm sure, the private sector. They have collaborations with different
Oh
groups.
yeah. Yeah, I mean the NIH, whatever the vaccines that
Yeah,
were developed for COVID was all based on NIH
yeah,
money, initially the basic research and development. Yeah, I
yeah,
see the Nobel Prize winners this year were involved in the early work on
yeah,
using mRNAs.
right.
See, the problem with kind of what we call naked mRNA, RNA, it's not very stable. There's enzymes all over the place called RNases that degrade it. So these people that got the Nobel Prize, they developed ways to chemically modify the RNA so that it can still be translated into protein, but it's not degraded easily. So when they inject it in, when it gets in your cells, when they administer the COVID vaccine, the mRNA for the spike protein gets in your cells and it stays around long enough so that the cells can produce enough of the spike protein and then spit it out so that your immune system can react to it. Yeah, that's
It's a small world. But when you were talking, it was making me think back to the Hormesis questions where you have like a rapidly degrading RNA, and then you're trying to find a way to modify that degrading process,
yeah,
and so I'm trying to, and then trying to just begin to wonder, how do you restore a degraded pathway, things of that nature.
yeah,
One of the questions that I've had, Mark, because I try to do different things, especially on, let's say, where you have an exercise mimetic or a caloric restriction mimetic, and plus I do exercise and I do intermittent fasting, and let's say you take berberine on top of
hmm.
on top of intermittent fasting. Is there? I know it has many different biological features, but I'm just kind of wondering, when you have multiple interventions on that are affecting the same sensor, so to speak,
Yeah,
maybe it just because what I've been doing that. I have multiple interventions, you might say, on the same aspect, and I if I were to look at my health status based upon measurements that people take or other things that go on, it seems like it's stable, if you know what I mean. It's not, it's they're all within the zone that's normal, whatever the readings are, and you say, well, at my age I'm functioning well, put it that way, and so I don't think that what I'm doing is harmful,
right.
that sort of thing.
Yeah,
It's and part of it is a lot of what I do, and these, like, with the so much emphasis on supplements and so forth, but a lot of these things just are poorly, they don't cross the blood-brain barrier well.
oh yeah, I think somebody, I don't take much at all. I take vitamin D and turmeric, and actually I'm taking vitamin K2 a couple of days a week too.
Yeah.
But otherwise I get my phytochemicals from the plants.
Yep.
I have a nice vegetable garden every year, and I'm, for I'd say 4 months out of the year, I'm eating vegetables from the garden,
Right.
and then in addition to that, I eat nuts and fish, and no sugars, and I do eat whole grains. I'm of the opinion that whole grains are actually fine. You'll find these talking heads out there and in the ether that say, oh, you shouldn't eat whole grain, you shouldn't eat wheat or whatever, but I don't have any problem with it. But so complex carbohydrates and then fruits and vegetables that have some of these natural phytochemicals in them, that's what I do. Otherwise I just take D and turmeric.
I've made such an effort to try to take a look at the hormetic effects of so many individual agents, from either the curcumin to the tea.
Well, I'm just going back to, this is what during evolution are the kinds of
Right.
toxins in the plants that we are normally exposed to. And so I'm just going that route.
Well, it's I think that this is an effective way to do it. It's very effective, and I think that it activates the systems that need to be activated. So it's good.
Yeah.
I just on a practical matter, I like your idea for a workshop. That'd be something that I would love to, maybe interact with you on to maybe help create.
Okay, let's follow up on that. We can come up with some like outline and then potential, participants. And, the other thing with this, it can work out really good because you can, have people can ask questions, either, verbally as we're going along and things like that.
No, this could be great, Mark. This could be it could be a whole series of these things. I mean, we could start something and
Yeah.
And just say, I mean, and if you have a concept.
Well, we'll get these entrepreneurs and celebrities on board too, and then they'll be able to
attract listeners.
put, like Sinclair and maybe Rhonda can, put the whole thing or links to it on their podcast.
And have them participate too. That would be great.
Yeah, no, I mean have them participate. Yeah.
No, that would be that's a great idea. I should probably let you go because we've been.
Yeah, I gotta go.
But this is
so I eat all my food during a 6-hour time window. It's going to be 5 or less today because right now when I'm done, I'm going to walk for a half hour and then do stationary bike for a half hour.
Excellent.
Yeah.
And probably 1:30 I'll head over to the UMass Rec Center and get tired for an hour.
Okay.
Okay. Hey, hang in there, Mark. Nice to see you, and this has been great. Appreciate it. You
likewise.
Okay,
take care. Bye.